So the first energy system that we use is the ATP PC system, also known as the PCr system. Just depends on your textbook and how it's referred to. So this is going to be anaerobic in nature, so it doesn't require oxygen. So this process is going to occur in the cytoplasm, not in the mitochondria. This particular process will produce a very little amount of ATP, and it doesn't last for very long. Usually within a couple seconds, the ATP for the system has been used. So it's one of the first ones that we use. It's super, super fast, and it doesn't last very long. Again, anaerobic in nature, so it happens in the cytoplasm, and we're going to talk about a little bit the physiology here. If you haven't watched the first lecture that talks about ATP, make sure you go back and review that. Otherwise, this whole process isn't going to make very much sense. So this is what our ATP PC or PCr system looks like. So here we've got phosphocreatine, an ADP, adenosine diphosphate, and then we're going to convert that into ATP or energy. Remember, ATP is what all of our food stuff gets broken down to create energy for our metabolic processes and other physiology of the body. And then we're left with the creatine left over. So what happens is we have an enzyme that comes in. So the enzyme is CK or creatine kinase. So creatine kinase is essentially going to steal this phosphate from phosphocreatine and donate it to our ADP. So as a result, we go from adenosine diphosphate to adenosine triphosphate, and then we're just left with the creatine because we stole this phosphate from it. So a kinase enzyme, so an enzyme you can kind of think of as the person that kicks the snowball that goes down the hill. An enzyme is going to be a catalyst that allows for a process to occur. So if we don't have creatine kinase, this isn't able to happen because it basically puts in the energy to get the system started. So a kinase enzyme, its role is basically to donate phosphates, or it steals phosphates, it transfers phosphates. So creatine kinase is going to come in here again and steal this phosphate from phosphocreatine, donate it to the ADP, and as a result, we're going to create ATP or energy, and we're left with a creatine.